Hi, my name's Janet and today we're going to talk about human immunity. The body has its own mechanism for defending against infection and disease, which include non-specific and specific defences. Non-specific defences include anatomic barriers such as skin, inhibitors, which slow or interfere with a chemical reaction, phagocytosis, which is a cellular process of ingesting cells, fever and inflammation. And then the specific defences, which are antibiotic defences against infection and disease. Just to recap, the body's three lines of defence include physical and chemical barriers, both non-specific, to prevent infection entry or a response such as inflammation, fever, phagocyte, complement proteins and cytokines to reduce and fight the infection, and the third line of defence is a specific defence, such as pus, swelling, T and B lymphocyte responses. Non-specific or innate defences exhibit the same reaction to all types of pathogens in the same way. They form defences that are present and effective from birth against a varied scope of pathogens or foreign bodies, such as a physiological response of inflammation, which increases the white blood count and at the location of infection, or a fever which increases core body temperature and inhibits further bacterial growth, ensuring the immune cells have a chance to fight the infection. Specific immunity involves the activation of B and T lymphocytes, which are white blood cells, which we will cover shortly. Here we're looking at the first lines of defence and the non-specific defences, which we have just covered. The first and second line of defence, first of all we'll look at the skin. The epidermis is the outermost layer of the skin, the body. First line of defence in a form of a waterproof barrier which protects internal organs from pathogens, chemicals, toxins and lower level radiation, and inhibits water loss to protect the internal organs. The skin enables this by continually renewing itself or healing with new cells, and makes melanin when exposed to strong sunlight, which is a substance that turns skin a darker shade to protect it from being burnt by the sun's ultraviolet rays. The dermis lies underneath the epidermis, and the subcutaneous layer forms the deepest section of the skin. The mucous membranes protect the surface of your eyes, ears, nose, sinuses, gastrointestinal tract, respiratory tract, mouth, throat, urogenital tract and vaginal tract. In the lining of the intestines, which provide a barrier against infection and aid digestion, the digestive tract contains barriers such as stomach acid and pancreatic enzymes, bile and intestinal secretions to protect against pathogens, and the contractions of the intestines and shedding of intestinal cells remove both harmful and dead microorganisms. The nose is a hotbed for microorganism entry from the external to the internal respiratory environment, but these can be filtered out through the nasal hairs or by being trapped by a mucus lining within the nose and the respiratory tract. Once trapped in the mucus, the pathogens can be removed by blowing the nose, coughing or sneezing. The bladder is protected by the urethra, which is a tube that drains urine from the body. Males are protected due to the length of the urethra, which prevents bacterial entry into the bladder, as well as constant urinal flushing away from any potential pathogens. However, insertion of catheters or surgical instruments can then introduce foreign substances and matter into that vulnerable area. Macrophages engulf and destroy pathogen particles. Their role is to detect, engulf and destroy any inert particles. They are specialised cells which constitute an important part of the body's defence in protecting the host from infection. Formed in response to dead or accumulated cells, the macrophages target and engulf cells before eliminating them. The cells are versatile and can transform themselves to form structures which can be more effectively fight specific infections. They are formed through differentiation, a type of white blood cell called monocytes. When there is an infection against damaged tissue present, monocytes will leave the bloodstream and enter the affected tissue. They undergo a series of changes to ultimately become macrophage cells. There are many varieties of macrophage with specific names according to where they function. A macrophage is a type of cell known as a phagocyte. Once infection has entered the first line of defence, for example the skin, often there will be an inflammatory response, which is a collection of cellular processes imperative to the immune system's response to infection or damage to the skin. The primary physical effect of inflammation is for blood vessels around the infection or damaged area to dilate and increase, creating inflammation. The second line of defence is the antibiotic response from cells, tissue and organs, which work simultaneously. The main symptoms of the inflammatory response are redness, heat, swelling and pain. Cells' walls around the affected area start to develop gaps which allow blood cells responding to the infection to pass through. As they are needed to respond to the infection, the tissue is infected will have a redness as a result of this due to the amount of blood circulating in this area. And the increased blood flow and immune system's response in the localised area of infection has several types of cells that assemble to contribute to the immune system. Because of the amount of blood and proteins that are attached to the infection, inflammation of that area is usual. This is due to a large quantity of proteins needed that fuel the immune response. The area will also become hot, which is an antibiotic response to infection. Acquired immunity is the third line of defence. Is learnt through exposure and response to antigens through its environment, whilst memorising the attack strategy and adapting itself for each specific antigen, either by naturally acquired immunity or through artificially acquired immunity, which can be developed artificially by either actively exposed to a vaccine or passively by reintroducing reformed antibiotics into the body for a protective response. Previously ineffective with the first exposure, antibodies are produced by stimulation after a foreign antigen is detected and the body remembers how to attack the antigen again through memory, when it's reintroduced, thereby acquiring stronger immunity. If vaccinated against a disease artificially, acquired immunity already recognises how to fight the antigen when exposed to it. The cells responsible for the acquired immunity are large B cells and T cells. Sensitivity to antigens, the role of different types of immune cells and hemoglobins, and there's white blood cells and leukocytes, which attack infections of foreign bodies, which come in 5 forms, all of which are produced in the bone marrow. Lymphocytes are white blood cells responsible for antibody production. The B lymphocytes make antibody and memory cells and that recollect a previous specific pathogen. T lymphocytes are responsible for cell-mediated immunity, and the third type of lymphocyte is called a natural killer cells, which are part of the innate immune system. The monocytes, which means that they can digest larger particles than other white cells due to their size when they leave the bloodstream. One of the largest white blood cells being in the okay, we'll move on to the next one. I'm not quite sure how to say this. Eosinophils combat against allergies and worm infestations. This is the cellular makeup of it. A little bit of information about it as well. We've also got, as part of the cells made in the bone marrow, the basophils, which secrete anticoagulants and antibodies to combat hypersensitivity and reactions. Here again is a little bit more information about the basophils. And then we've got the T cells, function by signalling for growth and activation of other T cells, macrophages and eosinophils. T cells, cytotoxic T cells, suppressor T cells and helper T cells all synchronise infection control, DNA damage and virus cells within the body. Cytotoxic T cells, suppressor T cells and helper T cells all synchronise infection control, DNA damage and virus cells within the body. And helper T cells aid regulation of immune functions by releasing lymphocytes, which then stimulate the T cells to grow or attack, as well as stimulating B cells to mature. Cytotoxic T cells defend against viruses by rejecting tissue grafts and form immune responses toward tumours. They're activated by the APCs. This is part of what happens when you have a rejection of blood or an implant. And they rely on the helper T cells and cytotoxic cell damage infected cells by attaching to them to use a protein porphyrin and release enzymes that eliminate the rest of the infected cell. Suppressor T cells stop helper and cytotoxic cells damaging the body's own tissue by suppressing them, which enable other cells to regulate the immune system. Suppressor T cells play a huge part in protecting against autoimmunity attacks. Again, these are the kind of cells that you want in your body when you've had a blood transfusion or an organ donation. B cells, humoral immunity. B cells provide antibody production, giving antigens to T cells and providing signals for T cell activation. Plasma cells are B cells that secrete antibodies into the circulatory system to maintain immunity. And memory B cells migrate to the lymph node after fighting infection, ready for the next invasion of that antigen species. Hemoglobins or antibodies are proteins bound to the antigens and generally fight infection. They enroll other immune cells, deactivate toxins, eliminate antigens, assist activation, spur phagocytosis of cells, activate mast cells and contribute to structure. The immune system recognises self and non-self through these antigens present and produced on all cells, and the differing cells enable the non-self to be attacked. The major histocompatibility complexes MHC1 are an early warning for the immune system if a foreign protein is detected, and MHC2 an extracellular presentation mechanism presenting antigens from pathogens by B cells and phagocytes, and the human leukocyte antigen gene complex help identify between self and non-self. If non-self T and B cells respond, sensitivity occurs from immune systems not having the same level of sensitivity to all antigens, and autoimmune diseases occur when cells react against self-antigens. This may initiate a lymphocyte response against cells which trigger autoimmunity when the antigens are totally unmatched and rejection occurs almost straight away. An example of this is when the wrong blood type is given. Here I've shown an example of rheumatoid arthritis, which is an autoimmune disease which affects joints, which down the road will affect mobility and function. We've got swelling and redness. And we've also got hypothyroidism, which affects the autoimmune disease, the thyroid, which produces hormones, and several areas of the body, which affects the metabolism, thin hair, foggy brain, the biology of vaccines. So, containing live or dead pathogens, fragments of the pathogen or toxins produced by pathogens, when exposed to the body's immune system for the first time, which is the primary response, these all act as antigens. The antigens are identified by B lymphocytes and according to the antibodies released by plasma cells. Memory cells are produced at the point which will recognise these pathogens if encountered again. Immunological memory is the immune system's ability to recognise a previous encounter with a pathogen and act more quickly second time around. When the pathogens are exposed to the body a second time, which is a secondary response, the memory cells act on producing the appropriate antibodies quickly and for longer, often before symptoms arise. Immunological memory are used to initiate memory cell production without the infection spreading. Vaccinations stimulate antibody production via white blood cells by the use of exposing the body to a harmless version of a pathogen to prevent future illness. Here we're looking at active immunity and passive immunity. So, natural immunity is acquired naturally from being exposed to the infection or disease through one's lifetime. Artificial immunity is injected antibodies containing preparation to initiate the antibody response. Passive is when a person is given antibiotics through blood or breast milk, and artificial triggers the immune response to produce antibodies. Viruses can be controlled by immunisation, as antibiotics don't work on viruses and still give us a good level of protection. The World Health Organization declared that smallpox has been eradicated in 1979 and Europe has been polio-free since 2002. According to World Health Organization, almost 4,500 deaths have been avoided by measles immunisation and 20 million infections have been avoided. In 1940, 857 deaths occurred from measles, and the following year it rose to 1,145. This compared to 1980, there were only 26 deaths from measles, and today we just have maybe one. So if you take into consideration the populations in 1940 and now, the benefits are huge. The basic concept of herd immunity: contagious diseases spread directly from person to person. There are a number of potential methods of transmission, which can be touching, bodily fluids, sneezing, airborne, waterborne. When a large portion of the population are vaccinated, it's harder to spread as there are very few people to become infected and the infection disappears. But when immunisation rates fall, herd immunity can be broken, leading to a rise in infections. There may be hundreds of variations of the illness. Pathogens may mutate, changing the antigens present. The microbes have different characteristics which affect their infection rate, some easily from person to person and others more contagiously. Other conditions are much worse in some demographic groups, all of which affect the threshold of vaccination necessary to provide herd immunity to the population of the whole world. And we've learned recently about the R number from Boris Johnson. If the R number is higher than the immunity threshold, has to be to protect using herd immunity. The threshold for measles is 94%. I'm not sure what COVID is. Therefore, it's important to roll out vaccination programs to capture a large amount of community. Otherwise, we cannot achieve herd immunity. Today, connections are global with mass production, and transportation means that diseases limits are global, and herd immunity through vaccination is critical. In the past, there's been some vaccination scares, especially one doctor believes that MMR vaccine caused autism in children. These studies have since been discredited, which today's field and anti-vaccination or anti-vaxxers who believe vaccinations cause cancer or cognitive decline, autism in some cases. Unfortunately, due to this, rubella is on the rise. I hope you enjoyed my video, and hopefully now you'll be able to understand more about specific immunity, the different kinds of white blood cells and how they all come together and synchronise an attack on an infection. Thank you. Bye.